Hi, I'm Susanna, and I'm here today to talk to you about the fly blood-brain barrier. But first, let's check the human blood-brain barrier. It is a layer of cells that protects, feeds, and cleans our brain. This layer is present at the brain capillaries. They are very tiny and thin blood vessels. Yet, if you were to stretch them all in a single line, you could go all the way from San Francisco to Portland. With such a huge structure, you can imagine how important this layer is to us. The endothelial cells, the pericytes, the astrocytes, and the extracellular matrix, secreted by all of these cell types, make up the blood-brain barrier. The endothelial cells are connected to each other by the tight junctions that physically seals the barrier. It excretes waste products from neurons and glia and important nutrients like vitamins, carbohydrates, amino acids, fatty acids, ions, hormones, and xenobiotics. They do so via specific transporters, receptors, and channels, and there are many thousands described so far. Thus, what goes in and out the brain is very tightly regulated. If the blood-brain barrier is damaged, nutrient uptake is compromised, toxic insults enter the brain and waste products accumulate, impairing brain function. Blood-brain barrier leakiness happens during aging, neurodegenerative diseases such as Parkinson's and Alzheimer's disease, and exposure to specific chemicals such as some immunosuppressants and TNB, an explosive present in munitions that are concentrated in some military installations. Study the basic mechanisms of blood-brain barrier formation and maintenance is crucial to understand how it gets disrupted and how to prevent or treat it. To study the blood-brain barrier in humans, we would bump into many ethical problems, difficult access, and years of wait. Because of that, alternative models to study the biology of the blood-brain barrier are needed. Guess what? The tiny fruit fly can be a good fit for this purpose. Although the fly doesn't have blood vessels, their entire nervous system is surrounded by a layer of cells that protects, feeds, and cleans the fly brain. This layer is composed of the extracellular matrix, the perineuroglial cells, the subperineuroglial cells, and the cortex glia. Although they have different names, these cells are very much alike the cells of the human blood-brain barrier. The extracellular matrix has the same composition as the matrix in humans, and the subperineuroglial cells are connected to each other by the septate junctions, just like the tight junctions. Moreover, this layer also excretes cellular byproducts and import nutrients via specific transporters, receptors, and channels. As well as in humans, their blood-brain barrier also breaks during aging, exposure to certain chemicals, and when researchers model human diseases in their brains. As if it wasn't enough, both humans and flies' blood-brain barrier have a circadian rhythm, meaning specific functions happen at specific times of the day. Both of them are responsive to nutrient levels in the blood or hemolymph, meaning they send to the brain specific signals that will trigger the activation of signaling pathways, which leads to neuronal differentiation and growth. Finally, because the blood-brain barrier tightly regulates what is delivered to the brain, it generally blocks xenobiotics, such as drugs that are being tested to treat several brain disorders. Thus, studying the blood-brain barrier in flies can also give us insight on how we can deliver drugs to the brain in a more efficient way.